Hello everyone, welcome back to the part 10 or chapter 10 of hematology, that is platelets. Consider subscribing to my channel for more of medical content and health tips. I'll be uploading my notes on Facebook as well as Instagram. Platelets. Platelets or thrombocytes are small, colorless, non-nucleated and moderately refractive bodies. So platelets are basically colorless, non-nucleated. Diameter is 2.5 micron and the volume of the platelets are 7.5 cubic micron. Let's understand shape. Platelets are spherical or rod shaped and they become oval or disc shaped when inactivated. Means when they are not activated, they are in oval or disc shape. Sometimes they even appear as dumbbell shape, comma shape, cigar shape or any unusual shape. Let's understand the structure and composition. So platelets have cell membrane, three structures, that is cell membrane, microtubules and cytoplasm. First, the cell membrane. Cell membrane is 6 nanometer thick. It has lipids, carbohydrates, proteins and glycoproteins. Together consist the cell membrane. Physiologically, some are important such as glycoprotein, which prevents adherence of platelets to normal endothelium but accelerate the adherence of platelets to collagen and damaged endothelium in the ruptured blood vessels. Next, the phospholipids, which accelerate the clotting reactions. Basically, cell membrane consists of all these substances which are physiologically important. Next, we come to the microtubules. Now, these microtubules, these form ring around the cytoplasm. These are made up of polymerized protein, that is tubulin, and give structural support to inactivated platelets and to maintain the oval or disc shape. Third part is the cytoplasm. The cytoplasm of the platelets include Golgi apparatus, endoplasmic reticulum, mitochondria, microtubules, and filaments and different types of granules. Proteins, enzymes, and hormonal substances, these are physiologically important and have particular functions. This is how the platelet structure appears. It has all the different cell organelles and it has the microtubules. Next we come to the normal count and variations. Platelet count is 2,50,000 cubic mm of blood. It can be 2 to 4 lakhs. That is the normal count of platelets. So basically it is physiological variation and pathological variation. First, let's understand physiological changes. Now in age, it is less in infants and by the third month it reaches normal from birth. Now sex, male and female have equal numbers except in menses female platelets decrease, and in high altitudes there is increase of platelet count, and after meals also the platelet increases. Pathological variations. It is very important. Thrombocytopenia, which means decrease in platelet count. Now this is seen in many such as acute infections, chickenpox, typhoid, tuberculosis, purpura and many similar diseases. Thrombocytosis. Increase in platelet count is called thrombocytosis. It is mainly seen in allergic conditions, asphyxia, hemorrhage, bone fracture, rheumatic fever and many such diseases. Third is the thrombocythemia, which is persistent and abnormal increase in the platelet count. So basically there is persistent abnormal increase such as in carcinoma, chronic leukemia and Hodgkin disease. So the three are thrombocytopenia is decreased platelet count, thrombocytosis is increased platelet count, and thrombocythemia is persistent and abnormal increase platelet count. Now we come to the properties of platelets. Here you need to remember the 3 A's, which are adhesiveness, aggregation and agglutination. So the 3A's, first we understand the adhesiveness. When having contact with any rough surface, the platelets are activated and they stick to the surface. Now the properties, the factors such as collagen, thrombin, adenosine diphosphate, calcium ions, von Willebrand factor, P-selectin, and vitronectin. These all are responsible for the adhesive property of platelets. Next is the aggregation. The activated platelets group together and become sticky. Now here for stickiness, ADP and thromboxane A2 are responsible. Third is the agglutination. Agglutination is clumping together of platelets. Now, due to the actions of some platelets, agglutinins and platelet activating factor, these are responsible for agglutination. So, don't forget the 3A's. Those are adhesiveness, aggregation and agglutination. Function of platelets. Let's understand the functions. Normally, the platelets are inactive and they execute their actions only when activated. So, they need activation for their functions. First one is role in blood clotting. Platelets are responsible for the formation of intrinsic prothrombin activator, which is in turn responsible for clotting onset. Second is role in clot formation. Now, in blood clot cells, platelets, these are entrapped in between the fibrin threads. Now, cytoplasm of platelets, these have particular contractile proteins such as actin, myosin, thrombocytin, which help in clot retraction. Retraction is breaking down of the clot. Role in prevention of the blood loss. Platelet accelerate hemostasis by secreting the 5-HT, which constriction of blood vessels takes place, where adhesive property and by formation of the temporary plug. Through this, it helps in blood loss prevention. Role in repair of ruptured blood vessels. Platelet derived growth factor formed in cytoplasm of platelets are useful to repair the endothelium. And in defense mechanism, by agglutination, platelets encircle the foreign bodies and destroy them by phagocytosis. Lifespan and fate of platelets. Lifespan is 10 days. It can vary from 8 to 11 days. Platelets are destroyed by a tissue macrophage system in spleen. Remember this, because whenever there is splenomegaly, platelet count decreases, and whenever there is splenectomy, there is increase in the platelet count. Thank you for watching my video. Do subscribe and keep supporting.